Hi students, welcome to chapter 3 of our cell biology course for medical laboratory scientists, and in this chapter we focus on DNA replication. So we know that DNA can undergo replication, and to replicate means to make more of itself. But why must DNA undergo replication? Well, remember that we have thousands of cells in our body, millions of cells in our body, billions of cells in our body, and each of these cells contains our DNA. Each of these cells contains all of our DNA. So when cells divide to produce more cells or to produce daughter cells, each of these daughter cells must contain the same DNA, and this DNA has to be in the same base sequence as the parent cell. Remember, the primary structure of DNA is the sequence, and so all DNA has to have the same sequence as the parent cell. The DNA therefore needs to undergo replication. So a precise copy of DNA is made. The reliability of DNA is very important, and replication therefore has high fidelity. So that means, as I mentioned, that each new cell that is made has the exact same copy with the same sequence of bases as the original cell. And fidelity is the degree of exactness with which something is copied or reproduced. Why do you think DNA replication must have high fidelity? Take a moment to think about it and see if you can answer this question. So DNA needs to have high fidelity because errors which could be introduced into the genetic code will pass on from generation to generation. And these errors could change the function or type of a protein produced. Remember that the sequence of the nucleotides or the sequence of the bases determines the type of protein that's produced. So if an error is introduced, you could introduce mutations, and these mutations can often result in disease because maybe the protein isn't functioning properly, like it's non-functional or it's over-functional or anything like that could result in disease. So what has to happen for DNA replication to occur? Remember, DNA is we learned about the secondary structure of DNA, and we know that DNA is a double helix. So the first thing that has to happen is that DNA needs to be unwound for replication, because what we want to see is, let me see, hold on if I can get a pointer. Here we go. If this is our DNA strand, we can see that it is a double helix. When DNA undergoes replication, it's these bases on the inside that are copied. So it has to open up to expose these bases so that replication can take place. Each of these new strands will then serve as a template, or each of these old strands, sorry, the original strands, will serve as a template for the two new daughter strands that are going to be made. And in this way, a precise copy of the DNA helix is made. DNA replication is very tightly controlled though, and so the process has a number of challenges. The first challenge is separating the two strands of DNA. We know that these bases are bound by hydrogen bonds, and they are complementary and they like to bind together. So the first thing that has to happen is these two strands have to unwind, and then we have to protect the unwound DNA from degradation. Okay, so there are enzymes that can degrade DNA. These are called nucleases. I'm sure you've heard of this already. And once your DNA is single-stranded, we have to protect it from degradation by these nucleases. Then another thing is that DNA can only be synthesized from the 5' to the 3' end. So remember, DNA is anti-parallel, right? So if, let's say, this blue strand starts at the 5' and ends at the 3', it means that the purple strand will start over here at the 3' and end on this side at the 5'. But the two strands have to be synthesized in the same direction, from 5' to 3' on anti-parallel templates. A little bit later, you'll probably understand a bit more what I mean by that. And then it's also very important that we are able to guard against errors in replication. As we said, so let's look at the first thing, separating the two strands of DNA. A helix destabilizing protein called helicase promotes the unwinding of double-stranded DNA. So within our body, we have helicase, which is an enzyme. You're going to need to know all the different enzymes that are responsible for the different processes that we're going to talk about. So for replication, the first enzyme that we're learning about is called DNA helicase. And this basically breaks the hydrogen bonds between the nucleotides and causes the unwinding or separation of the DNA strands. Now, as we said, once the strands are single-stranded, once the DNA has unwound and separated, the single strands are in danger of being degraded by nucleases, right? So now we have other proteins called single-stranded binding proteins, which are these little guys here, that will bind to the single-stranded DNA to protect it from degradation by nucleases. Another function of your single-stranded DNA binding protein is that it prevents the single strands from reannealing to form double-stranded DNA. Because remember, these bases are still complementary. They want to bind together. They like each other. They have high affinity for each other. And so the single-stranded binding protein prevents the reannealing of the two strands. Then we also said that DNA needs to be synthesized from the 5' to the 3' end. Okay, so DNA needs to be synthesized on each of these two strands because each of these strands act as templates to make two new daughter strands, and it can only take place in the 5' to in the 5' to 3' direction, complementary to the parent strand. So it looks something like this. If this is the 3' end, it means that this end would be 5' of the new strand, and it can go from 5' to 3'. And on this strand, it will go in this direction, right? 5' to 3'. But to understand how this takes place, we need to understand that DNA replicates by semi-conservative replication, by bidirectional replication, and by semi-discontinuous replication. So these are very important terms that you need to know and you need to be able to explain. At the moment, they may sound a bit confusing, but we're going to go into detail so that you can understand what each of them means. Let's look first at semi-conservative replication. We know that DNA involves the separation of two strands, right? And the new strands are synthesized using the original strands as templates. So they will look at the original strand and it will be copied. So the original strand is used as the template. Each new daughter molecule will then contain one strand of the original DNA molecule, so which you can see here in blue, and one new strand. This is called semi-conservative because it is half conserved, right? The original strand, it has one old and one new, so it is half conserved. So therefore it's called semi-conservative. Just to clarify, if you're not sure about what the word conserve means, to conserve is to save. So you haven't used the old DNA and then discarded it. It's still saved within the new daughter molecule. DNA replication is also bidirectional. What does this mean? Well, it means that DNA is synthesized in two directions. So remember, the DNA helix is unwound by helicase, but this unwinding happens at a point called the origin of replication, REC for short. Okay. The origin of replication is a specific position on the DNA that will tell the helicase where to unwind. So if over here on this figure, this is your origin of replication. For each origin of replication, there are two replication forks. So this is one of the forks, and this is one of the forks. You can see the DNA forks because it splits here. And it's at these forks that the new nucleotide chains are formed. So new strands of DNA are synthesized from the two exposed strands, which we heard about just now. but it happens in two directions, okay? Always from 5' to 3'. So DNA has to form from 5' to 3'. And this area over here between the two forks is called the replication bubble. To show a picture of what bidirectional replication looks like, you have your origin of replication where your DNA will split in two. Okay. This is your replication fork because this is where the DNA is splitting. And new DNA will always form in the 5' to the 3' direction. Like this. So it goes in this direction, but it also goes in this direction. So this means that it's bidirectional. Prokaryotes have got circular DNA, so there is one origin of replication and one replication bubble. However, in eukaryotes, remember we have got lots more DNA, and so there are several origins of replication and several replication bubbles. So if this is a strand of eukaryotic DNA, you can have a number of different origins of replication, and the bubbles you'll have a number of different replication bubbles, and the replication forks will move towards each other until they meet, and then they will merge and you will have two daughter DNAs. So you can see it here. Here's your replication bubble, which gets bigger and bigger, and eventually the two merge and you end up with two molecules. Where in circular DNA, you'll have one origin of replication, and it will be like the circle is splitting in two and forming two new daughter molecules. Then DNA is also undergoes semi-discontinuous replication. So remember, these three terms are all describing the process of replication. They're not three separate things that happen. They're just describing this one process, right? So as we said, all DNA is synthesized in the 5' to 3' direction. And we know, we've looked at this before, we know that it is 5' because there is a free 5' carbon at this end, and there is a free 3' carbon at this end, right? So DNA can only grow in this direction with a new nucleotide being added on this end the whole time. Nucleotides will never be added onto the 5' end. They will always only be added onto the 3' end. And this hydroxyl group on the 3' carbon is known as a nucleophile. Remember, something that's a phile means that it's loving, so it is a nucleotide loving area. But now, how does replication take place if the two strands are going in opposite directions? Remember, it can only take place from 5' to 3'. But it has to take place on both strands. So on the one strand, the leading strand, your DNA can very easily form from the 5' to the 3' because as the replication fork moves and more nucleotides are exposed, it can just add more bases onto the 3' end. That's simple enough. But on this strand that we see here, the 5' end is not exposed all at one go, right? So it has to form in fragments. This is called the lagging strand, and it is formed semi-discontinuously. This is the leading strand, and because it can be made in one go, this is formed continuously. Okay, this is formed discontinuously. It's formed in small fragments called Okazaki fragments. The 5' end of the new daughter strand is closer to the replication fork. And then after all of this, the DNA fragments will be joined together by an enzyme called DNA ligase. So just to round up for this segment, DNA replication is semi-conservative. Remember, it contains one new and one old strand. It is bidirectional. DNA always is formed from the 5' to the 3' direction, but it takes place in two directions, and it is semi-discontinuous. The leading strand is formed continuously. The lagging strand is formed discontinuously. So that means it is semi, which kind of means half discontinuous.